Hello, everyone, and welcome to today's Model Aquatic Health Code Network webinar. It's a pleasure to have you all here. My name is Daisy Galan. I'm a lead program analyst at the National Association of County and City Health Officials, NACHO, and I will be moderating today's webinar together here with my colleague, Rebecca Ryan. This webinar is being recorded, and I will be shared along with the presentation slides following today's event. Please feel free to submit any questions that you have via our Q&A box throughout the webinar. We'll address all your questions at the end. Next slide, please. With that, we'll start with today's agenda. We're gonna start with a few updates from CDC. And then we'll move to our presentation with our guest speaker, Dr. Blatchley, and then we'll have a session at the end for questions and answers. With that, I will pass the word to Joe Lako from CDC, who will share some announcements with us.
Hey, good afternoon, everybody. I really appreciate you spending a little bit of your time today. We have an exciting webinar scheduled for you. To be nice and technical. Haven't had one of these in a while, so we're excited. I did want to let everybody know that CDC has finalized the 4th edition of the Model Aquatic Health Code. We got it through clearance, and we've made it accessible with 508 compliance, and are currently working to get it up on the Healthy Swimming website, the cdc.gov website. If anyone is in urgent need of it or would like to have a peek at it before it is on the website, please feel free to reach out to me. I can email those PDFs of the 508 documents to you. But that was our big announcement for today. I wanted to let everybody know we've wrapped up the 4th edition MAC, and we're already beginning to think about the 5th edition and the change request that'll be happening later this year. Thanks, Daisy.
Thank you, Joe. And with that, it is my pleasure to introduce today's speaker. We are very happy and honored to have Dr. Ernest Blatchley here with us. Dr. Blatchley is a Lee A. Riff Professor in Environmental Engineering at Purdue University, where he works a joint faculty appointment in the Lyle School of Civil Engineering and the Division of Environmental and Ecological Engineering. Professor Blatchley conducts research in a broad area of physical-chemical processes of environmental engineering. The chemistry and dynamic behaviors of swimming pool, water, and air have been focal points of research in his group for roughly 20 years. With that, thank you so much for joining us today, Dr. Blatchley, and we'll pass it on the floor to you.
Great, thank you, Daisy, for the introduction. Okay, can you see my screen now? Hang on for just a second. Can you see my screen? It should be title slide.
Yes.
You can hear me, I assume?
Yes, perfect.
Okay, great. All right, so I'm going to be talking today about some research we've been conducting in my group for a few years that addresses the dynamics of gas phase trichloramine in indoor swimming pool facilities. Before I get into sort of the gory details of this presentation, I want to acknowledge some of my collaborators. They include Brandon Bohr, a faculty colleague of mine here at Purdue, and one of his graduate students, Danielle Wagner, Antonios Tasoglu, and then in the image on the right, there are 4 collaborators who played crucial roles in these experiments and numerical simulations that I'm going to present. First is Lester Lee, who's here using an instrument called a portable MIMS device. I'll talk about that device later in this presentation. In the background are 2 other students, Tianren Wu and Jinling Jiang, who conducted measurements using a really high-end instrument that we were fortunate to be able to configure at a pool called a proton transfer reaction time of flight mass spectrometer, basically a fancy instrument for doing detailed measurements of compounds in the gas phase. And then last and certainly not least, on the right is Thomas Fuldis, who developed a device called an online derivatization cavity ring down spectrometer, or ODCRDS, This is a really cool device that allows for real-time measurements of gas phase trichloramine. It's a unique instrument, and we were very fortunate to have access to it and Thomas for some of the experiments that I'll describe here. All right, so the focus of this presentation is going to be on trichloramine. So let's just get to some of the basic characteristics of trichloramine. For various reasons, some of which I'll describe, I view this as sort of a sentinel compound for indoor air quality, or IAQ, in indoor pool facilities. It is produced as a result of a number of reactions that we know take place when free chlorine comes in contact with the reduced nitrogen compounds that are introduced to swimming pool water by swimmers. Those include urea, uric acid, creatinine, and a range of amino acids. This compound, trichloramine, is also responsible for the characteristic chlorine odor that we associate with indoor swimming pool facilities, along with a structurally related compound that's listed here that's identified as dichloromethylamine. It causes irritation of various human tissues, including the respiratory system and eyes. And with some people, trichloramine and the other chloramines also cause irritation of the skin. And then last, and certainly not least, from a structural or sort of infrastructure perspective, trichloramine is an important contributor to corrosion that we see in indoor swimming pool facilities, not only on the stainless steel and conventional steel surfaces that are illustrated here, but also within the ductwork of HVAC systems. So let's talk for just a minute about volatile compounds in pools more broadly. So trichloramine is a volatile compound. So what does that mean? Volatility is defined as the ability of a compound to be transferred from the liquid phase to the gas phase. So what that means is that volatile compounds that are present in water, like in a swimming pool, have the potential to affect indoor air quality if there are processes that promote their transfer from the liquid phase to the gas phase. And we'll be focusing on that in this presentation. One measure of volatility is the so-called Henry's law constant, or capital H. And that value, the larger that value, the more volatile a compound is. So generally, the larger the Henry's law constant, here represented in units of atmospheres in the right-hand column, the more volatile the compound is. So it turns out that in terms of the mechanics of transfer of compounds from the liquid phase to the gas phase, the compounds that tend to have the largest Henry's law constants, or in other words, those that are most volatile, meaning having a Henry's constant greater than something like 10 to 50 atmospheres, the physics that are responsible for transfer of those compounds from the liquid phase to the gas phase are identical. And that's because they are so volatile. So it's also important to point out that there's another volatile compound that's present in all swimming pool waters, and that is carbon dioxide. It also has a relatively large Henry's law constant. And as we'll see, that large Henry's law constant for carbon dioxide and the fact that it's present in all water samples suggests that carbon dioxide might be a fairly useful proxy for these other compounds that generally tend to be more difficult to measure in the gas phase. So one other attribute of these compounds that have relatively large Henry's law constants, or that are relatively volatile, is that their transfer is going to be governed by mixing of water near the air-water interface. So practically what that means is that anything that we do in a swimming pool that promotes mixing of water close to the air-water interface is going to promote transfer of these compounds from the liquid phase to the gas phase, and therefore that will affect indoor air quality. The central hypothesis of the work that I'm going to present is that with all other conditions being constant, the worst air quality that we're going to observe in an indoor swimming pool facility or natatorium will occur at the times of heaviest bather loading. There may be other attributes that contribute to this, but this one seems pretty clear. To address this hypothesis, we conducted a series of measurements at swimming pools where we measured various aspects of water chemistry and air chemistry before, during, and after periods of both normal use and heavy use. And these measurements were all conducted in lap swimming pools, conventional, back and forth kind of lap swimming pools, and oftentimes before, during, and after relatively large competitions. We also measured a number of other factors that we believe have a role to play with respect to indoor air quality dynamics, including the characteristics and behavior of the HVAC systems, some attributes of pool operations, the number of swimmers. Sometimes we care also about the number of people who are on the pool deck or in the bleachers. The bottom line is that we identified the factors that we believe are relevant with respect to these indoor air quality dynamics, and we quantified them. We used the information from these experiments to develop a mathematical model. That mathematical model allows us to numerically simulate this dynamic behavior that we care about. So these numerical simulations, or this modeling that I'm going to talk about in the second half of this presentation, is based on a conventional mass balance approach. And in that approach, we needed a submodel to describe the dynamics of transfer of these chemicals from the liquid phase to the gas phase. And we used what's known as two-film theory that I'll describe briefly at the end of or in the second half of this presentation to describe those dynamics. And ultimately, we use these models then to simulate the overall dynamic behavior of trichloramine and, as I'll show you, carbon dioxide in the gas phase. So this project that I'm going to describe is a three-phase project. What I'm going to present today are the results of phases 1 and 2, which have focused on competition pools located in the upper Midwest. And that's largely just for convenience from our perspective. They're close to where my lab and where my students are. In phase 3 that I'll describe briefly here and later on in this presentation, what we hope to do are to conduct similar experiments and simulations in other geographic areas and with other facility types. So let me spend a minute or two talking about the materials and methods that we applied here. So for measurement of liquid phase or aqueous trichloramine and other volatile disinfection byproducts, we used a method called membrane introduction mass spectrometry, or MIMS, and I'll talk about that more in just a minute. For quantification of liquid phase or aqueous carbon dioxide, we used measurements of alkalinity and pH, and then some equilibrium calculations to allow us to estimate this number over here, the concentration of CO2 in water. We used a device called NEMO that was provided to us by a company in France to allow real-time measurements of gas phase trichloramine, gas phase relative humidity, and gas phase carbon dioxide. For one of the experiments that I'll talk about, and really will be a focus of what I talk about here, we used these pretty high-end instruments that we were able to configure on the pool deck at a pretty large competition. They include this online derivatization cavity ring down instrument that Thomas Foldis brought to these experiments, the time of flight mass spectrometer, and then another instrument called a LICOR. It's made by a company called LICOR that allows for real-time measurements of carbon dioxide in the gas phase. Along with the lifeguards in the facilities, we counted the number of swimmers who were in the pool as a function of time. We also quantified the number of people who were on the pool deck and in the bleachers as appropriate. We did measurements of airflow through the HVAC system. And again, these are all the parameters that we cared about and that we believed played a role in terms of influencing the dynamics of these chemicals in the gas phase. Now, our focus was on trichloramine and carbon dioxide. Trichloramine for reasons that I've already described in terms of its characteristics, largely nuisance characteristics in swimming pools, and CO2 because, frankly, it's much easier and less expensive to measure, and it behaves in a manner that's similar, though not identical, to trichloramine. The experiments that I'm going to present and the corresponding simulations are all based on venues that are located in Indiana and Michigan. Again, that's for convenience. 5 of these were at one pool that happens to be located in Indiana. A 6th experiment was conducted at a high school pool also located in Indiana. And then there were 2 experiments that were conducted at university pools in Michigan. These are all lap pools, and the experiments that I'll describe were conducted before, during, and after periods of either normal use or heavy use. And the heavy use circumstances that we evaluated were large competitions, swimming meets. All right, so I mentioned this method that's called membrane introduction mass spectrometry, or MIMS. So here's a brief description. I'm hoping that you can see the cursor. So I'm floating the cursor around this green arrow that describes where we introduce a liquid sample. The idea is that this liquid sample is going to be introduced to this tube. The sample is going to be pumped through this tube where it will then encounter this device that we call a per-evaporation membrane. It's another tube, and it's a tube that's made of just simple silicone. So the silicone itself will reject water, meaning the water will not be able to go through the membrane, but volatile compounds, the ones that we care about, will diffuse through the wall of the membrane and then go into the other side or go across the membrane to the other side. And then what we're going to do is introduce an inert carrier gas, nitrogen or helium, and we're going to sweep those compounds into a mass spectrometer. That allows some important benefits. First of all, mass spectrometers are very sensitive. Limits of detection for these things are pretty low relative to other methods. And second, because we're using a mass spec for analysis, it also allows identification of the structure of these compounds. In addition to those measurements, we also conducted measurements using this device that I mentioned briefly called NEMO that was provided to us by Ithera Labs located in France. And we had several of these NEMO devices that allowed for measurements of gas phase trichloramine, but also measurements of relative humidity and carbon dioxide in the gas phase. And then lastly, we used this LICOR instrument to conduct real-time measurements of gas phase CO2. So those are the measurements that we conducted. This is a table, and I'm not going to go through all of this. The point really is just to illustrate that we conducted a number of these experiments at different facilities. The facilities were not identical in terms of their geometry or how they treat water or how they were used, or for that matter, the nature of the swimmers who were in the pool at the time of the experiments. So the idea was to provide fairly broad coverage of circumstances that would be representative of these lap swimming pools. And we think we've accomplished that, at least to the extent that was possible with the funds that were available in phases 1 and 2. All right, so this is a complicated slide. I'm going to spend a couple minutes on it to just sort of walk you through this because I'm going to show a couple slides that look like this. So the horizontal axis is time that represents days and then hours through the day. The left vertical axis represents gas phase trichloramine concentration, and that corresponds with the the green and red traces that I'm following with the cursor right now. And those illustrate the dynamic behavior of gas phase trichloramine. The dashed, I guess this is pink and blue lines, represent gas phase carbon dioxide. And then the vertical bars represent the number of people that were present in the facility, either in the water or on the pool deck. So there's several trends that show up. First, this was an experiment that was conducted with 8 and under swimmers. So these are small children at a competition. There were about 350 swimmers who were present at this time, and there were as many as 150 or 175 children swimming laps in the pool at one time. So a pretty crowded pool typically during the warm up and warm down periods of this event. Another thing that's a very consistent trend that we observed in not only this experiment, but all of the other experiments is that there's a really strong correlation between the times when people are present in the pool and when we see peaks in the concentrations of these compounds that we're measuring in the gas phase. And the reason for that is really pretty simple. It's because the swimmers who are spending their time right near the gas liquid interface are mixing that water and promoting transfer of these volatile compounds. So when you put 150 small children in a pool and ask them to swim, they're going to be mixing that water and promoting transfer of those volatile chemicals from the liquid phase to the gas phase. What that means is that the concentration of those compounds in the gas phase is going to go up. So that's what we observed, and that's a consistent trend that shows up in this data set, and in fact, in all of the data sets that we've collected. So here's a different experiment. This experiment was conducted in the same pool facility, but now with adult swimmers. These were actually elite swimmers, and there were up to 200 of these swimmers that were present in the pool at any point in time. Again, what we observe is a very strong relationship between the number of people who are present in the pool and these peaks that show up in the concentration of gas phase trichloramine or gas phase carbon dioxide. Moreover, the shapes of these peaks and the timing of these peaks, both for carbon dioxide and for trichloramine, are almost identical. They match each other quite well. And I'll show you more data to illustrate this later. Here what I've done is just separate out a little bit of the data. This is data that were collected using the LICOR instrument. And let me just focus on this instrument and how we conducted these measurements. At the top of the legend here, you'll see that there are 4 locations: bulk air, surface air, return air, and supply air. So with this particular experiment, we had one of these LICOR instruments, but we set it up so that it could collect samples from 4 different locations. The way that we did that was we had sample tubing that was connected to a multi-port valve, and that multi-port valve was set up to automatically rotate between these various positions. So we had a schedule of about maybe 30 minutes long, where during that 30-minute cycle, this 4-position valve would rotate between bulk air, surface air, return air, and supply air. And the different colors that are evident in this series of data that I'm following with the cursor right now follow a consistent trend, which supports the idea that the airspace in this facility was well mixed. We have other data that support that as well, but that's certainly one piece of evidence to support the well mixed assumption. Perhaps more importantly, what this illustrates, again, is this very strong relationship between peaks in the gas phase concentration of a volatile compound, in this case CO2, and the number of people that are present in the facility. In the case of CO2, we need to account for people who are in the water and people who are on the pool deck for reasons that I'll describe in a couple minutes. All right, here's another illustration of a similar data set. This is basically from the same experiment. And there's a number of lessons that we learn here, but the lessons really that are learned by this much more detailed data set are essentially identical to those that I've already described. So the bottom line is that we've identified through observations a very strong link between gas phase trichloramine and the number of people who are present in the pool swimming. We have also identified a similarly strong link between gas phase concentration and the overall number of people who are present in the facility, including swimmers and non-swimmers. Lastly, it's difficult to see on this slide, but I'll illustrate it on the next one. We did see evidence of what's sometimes referred to as the bubble effect. So let me just explain for those of you who may not have heard this term. The bubble effect is used to describe a phenomenon where the concentration of a volatile compound will be substantially higher just above the air-water interface, in other words, right above the pool, than it would be farther up above that pool. So what we observed was that this bubble effect was measurable and evident only during periods of relatively heavy use, and that it was quite temporary. So let me show you what that means. So in this data set, which is exactly the same as the data set that I showed on the previous slide, it's just confined to a much shorter period of time, here roughly 6 hours. What I want to focus on are these differences in color between these panels. So this greenish color that I'm pointing to with the cursor right now represents when samples were being collected from what we refer to as the bulk air. That bulk air sample was collected from a position 3.2 meters above the pool deck, so about 10 feet above the pool deck. In the next panel, immediately to the right, this pinkish color, then that 4-position valve was switched so that we were collecting air samples from 40 centimeters above the pool, so a little bit more than 1 foot above the surface of the pool. So when we switch from this location, 10 feet above the pool, to this location, 1 foot above the pool, basically in the same period of time, we see this pretty substantial shift upward in the concentration. And what that's telling us is that in this period of time, when there were a lot of people who were present in the pool, we see evidence of this bubble effect. Concentration just above the pool surface is higher than it is in the bulk air. And this wasn't the only time when we observed this. You can see this over here as well. But pretty much what we saw was that there was evidence of this bubble effect only when the pool was occupied by a lot of people. In other words, when it was really crowded. And that bubble effect really dissipated relatively quickly. As soon as those swimmers got out of the pool, the bubble effect went away. All right, so on this slide, what I'm showing are correlations between what we measured in terms of relative humidity and the concentration of trichloramine. So we measured trichloramine using several different analytical methods. There are two that are illustrated on the horizontal axes of these graphs. What we're showing here is that relative humidity and trichloramine concentration do correlate pretty strongly. So what that suggests is that the conditions that promote the transfer of water vapor from the pool to the air above the pool are the same as the conditions that are going to promote transfer of trichloramine. And likewise, what we observe in these bottom two graphs is that the conditions that promote transfer of carbon dioxide are the same as the conditions that promote the transfer of trichloramine. In other words, what we observe are strong correlations between gas phase trichloramine and gas phase CO2. That supports the use of CO2 as this proxy that I mentioned before. We also see strong correlations between gas phase trichloramine and relative humidity, which suggests that it might be possible to use relative humidity as a proxy as well. All right, so generally, what are the lessons that we've learned from these field experiments? First, indoor air quality in indoor swimming pool facilities is going to be affected by water chemistry. Water chemistry, in turn, is going to be affected by swimmer hygiene. Do they take a shower before they get in the pool? Do they pee while they're in the pool? Those are both very important questions, and they have a clear influence on water chemistry. Water chemistry is also going to be influenced by how we treat the water in a swimming pool facility, pH, alkalinity, and a number of other factors. Indoor air quality is also influenced by swimmer number and activity, as well as the characteristics of the HVAC system. There's also a strong link between swimmer number and indoor air quality. Generally, the worst air quality coincides with the conditions where we have the largest number of people who are present in the pool. And as we'll see, it's not just the number of people in the pool, it's also the characteristics of those swimmers. So if we put adult elite level swimmers in the pool, that's going to be very different than putting 8-year-olds in the pool in terms of their ability to transfer volatile compounds from the liquid phase to the gas phase. And we'll see that in a few minutes. Lastly, carbon dioxide appears to have characteristics that suggest that it might be a reasonable surrogate or proxy for trichloramine. And so measurements of carbon dioxide might be relevant as a useful substitute for those measurements of trichloramine. All right, so what I'm going to do for a few minutes is talk a little bit about the physics that are involved in the transfer of a compound from the liquid phase to the gas phase. And I'll use trichloramine as an example. So the way that I'm going to describe this is using the logic that's presented in the so-called two-film model, which was developed a long time ago and really still represents a standard for descriptions of these sort of dynamic behaviors. So imagine that we have a molecule down here, trichloramine, that's present in the water, so it's aqueous. What we're interested in is how does that molecule get to the gas phase? In the two-film model, the idea is we imagine that there are going to be a series of steps that this molecule is going to have to take. The first is going to be to move from what I'll refer to as the bulk water down here to the edge of what's oftentimes described as a liquid film. That liquid film is assumed to exist only right next to the gas-liquid interface, and it's assumed to be static, which means that transfer of the molecule from or across that liquid film is going to be by diffusion, a relatively slow process. We assume that there's a similar film on the other side of that interface, and that transfer of a molecule across that film will also be by diffusion, but now in the gas phase. And once that molecule then reaches the edge of that gas film, then mixing behavior that might exist in the gas phase will cause relatively rapid transfer of that molecule. So we have these 4 steps that take place in sequence. And the idea in the two-film model is that the overall rate of transfer is going to be defined, it's going to be governed by a rate limiting step. In other words, the idea is that one or perhaps two of these processes are going to be very slow relative to the others. And the logical candidates are going to be diffusion across the liquid film, diffusion across the gas film, or both. So staying with the idea of sort of resistances in series, sort of like we do in electrical engineering, what we're going to do is represent the overall resistance, that's the left side of this equation, as the inverse of what we call a mass transfer coefficient. So a mass transfer coefficient defines the ability of a system to move a molecule from the liquid phase to the gas phase. The larger that value is, the lower the resistance is, and the faster the transfer will take place. We can represent that overall resistance then as the sum of two resistances, the resistance that's offered by the liquid film and the resistance that's offered by the gas film. But you'll notice in this gas film resistance term that I'm pointing here to this capital H in the denominator. That is the Henry's law constant that I talked about before. So when this Henry's law constant is a relatively large number, that makes this whole ratio here small, which means that the overall resistance, the number on the left side, under those circumstances will be dominated by liquid film resistance. So anything that we can do to reduce the resistances offered on the liquid side of that interface will promote transfer. So for example, what we could do is introduce a swimmer. A swimmer will promote mixing. And so there's much more circulation that's going to take place on the liquid side of that interface as a result of a swimmer being present. The effect of that is to reduce the thickness of that liquid film. And because of that, that increases the rate of diffusion or the rate of transport by diffusion across that liquid film. So this mixing of the liquid phase then plays a key role in governing the transport of these volatile chemicals. And again, the idea is that we're going to focus on this, sorry about that, we're going to focus on this liquid film resistance, and we assume that because we're dealing with compounds that have relatively large Henry's law constants, this term, which describes gas film resistance, goes away. It doesn't contribute at all. All right, so if we evaluate the fraction of the overall resistance that is offered by the liquid film, that's what's represented on the vertical axis using a log scale here, what we observe is that it's strongly dependent on the value of that Henry's law constant if everything else is the same. And as I mentioned previously, if we have compounds that have relatively large Henry's law constants, then the overall resistance is dominated by the liquid film. And if that's the case, then the mechanics of transfer are going to be the same. So if you look at the list of chemicals that are present, that are common in swimming pools, and evaluate their Henry's law constants, what we find is that trichloramine is well to the right of this sort of vertical dashed line where we observe liquid film control. In other words, any compound located to the right of that vertical green dashed line is going to be dominated by liquid film control. But we also see that CO2 has this relatively large Henry's law constant, so we expect its behavior to be very similar. And in fact, it is. All right, to build on this just a little bit more, this is a table in which I've described several compounds listed on the left that are common to swimming pool water. And here I presented typical liquid phase concentrations. The next column over here represents their Henry's law constants. And so we can then calculate the concentration in the gas phase that would be in equilibrium with this liquid phase concentration that I've listed over here. And then the last column presents the relatively few measurements of the gas phase concentration of these compounds that have been reported in the literature. So there's some common lessons that show up from this. First, there are several of these disinfection byproducts that have relatively large values of Henry's law constants. And from that, that suggests that their physics of transfer will be the same. In other words, if we develop a model to describe the transfer of trichloramine, we should expect that model to work for the other volatile compounds as well. Next, if you compare the reported gas phase concentrations in this last column with the equilibrium concentrations that are over here, what we observe is that the reported values, or these observed concentrations, are typically very small relative to equilibrium. That turns out to be important in the modeling because it turns out that we never get close to an equilibrium condition. In fact, with many of these chemicals, if we ever did get close to an equilibrium condition, like with trichloramine or cyanogen chloride, it would be deadly in terms of that air quality. So it's good that we never get close to that. But it also has implications in terms of how we're going to simulate these dynamics. So this is a schematic illustration of the model that we've developed to describe these dynamic behaviors. This is specific for trichloramine. So let me just walk you through this. This is supposed to represent an indoor swimming pool. The idea is that the HVAC system will bring in air from the outside at some flow rate that we'll call Q outside. That air is going to go through a heating, cooling, and dehumidification process and then be recirculated. And the air will leave the facility at the same volumetric flow rate, although its composition will be quite different. This symbol over here represents the mixing that is inherent in the pool water that results just from recirculation of water through the treatment system. So as the water circulates like this through the treatment system, there's going to be some mixing that takes place. That mixing of the water will then promote some transfer of volatile chemicals like trichloramine. But then we're going to put swimmers in the water. The swimmers do at least two things for us that are relevant. First, they're going to introduce precursors. Those precursors are going to react with chlorine, and they're going to generate the disinfection byproducts, including trichloramine. And second, they're going to mix the water, and as it turns out, much more aggressively than the ambient circulation just from sending water through the treatment system. And that also is going to promote transfer of trichloramine. So when we develop a model to describe the dynamic behavior of trichloramine, we need to account for both of these emission processes of trichloramine from the water in the pool to the air above the pool. So mathematically, this is the model that we use to describe these dynamics. And I think what I'm going to do in the interest of time and perhaps the sanity of the people who are listening is I'm going to skip through the math here pretty quickly. I'm happy to try to answer questions related to this if they come up. But the basic idea with this model is that we're going to use this model to simulate how the amount of trichloramine that's present in the air above the pool, that's what's described on the left side, is going to be influenced by the various processes that are present in the system. Those processes include the introduction of outdoor air, the air being exhausted from the facility, the baseline flux of this material from the water to the air just as a result of circulation of water through the treatment system, and the promotion of transfer by the swimmers themselves, the activity of the swimmers. All right, so again, what I'm going to do is skip through this. In fact, I think in the interest of time and sanity, I'm just going to go through this very quickly. Again, if there are questions about this, I'm happy to go through the details of the math. But what I'm going to do is just move to this slide to explain sort of the physics and chemistry that are relevant with respect to CO2. So imagine that we have some CO2 that's present in the air above a pool. That CO2 will come to equilibrium, or at least it will dissolve in the water, and it will form aqueous CO2, which will then react with H2O to form carbonic acid. That in turn will dissociate to form bicarbonate, which in turn will dissociate to form the carbonate ion. So we understand this chemistry in detail. If we have water being circulated through a treatment system as we considered previously, there will be some CO2 that will be transferred from the liquid phase to the gas phase as a result of that mixing process. And if we stick a swimmer in the water, again, that swimmer is going to promote transfer of this volatile chemical that we call carbon dioxide for the same reasons that it'll promote transfer of trichloramine. But in the case of carbon dioxide, we need to also consider the CO2 that's being released by the swimmer as a result of just natural respiration. We also need to account for people that are non-swimmers and their respiratory activity. So in the model that we developed to account for carbon dioxide dynamics, we accounted for the same things that we accounted for in the trichloramine model, but we added terms to account for the emission of carbon dioxide by the swimmers just through respiration, and a similar term to describe the emission of carbon dioxide by the non-swimmers in the facility, because they contribute significantly to the amount of CO2 that's present in the system. Okay, Again, in the interest of sanity, I'm going to move through this relatively quickly, but I'm happy to come back to it if there are questions. What I want to do next is describe, or let's say compare, carbon dioxide emission rates from swimmers and non-swimmers. So here is information that we collected by a survey of the literature that describes the behavior of swimmers and non-swimmers. And let me just walk you through this. So what I'm presenting here are emission rates for individuals, single person, either a swimmer or a non-swimmer. And what's represented in these equations are, first of all, the so-called tidal volume. That's the volume of air that you either breathe in or breathe out per breath. And what we're comparing is that volume for a swimmer versus a non-swimmer. Not surprisingly, people who are exercising aggressively like swimmers are going to take in much more air per breath than a non-swimmer. And here's a comparison it's about roughly 4 times as much. They will also be breathing much faster. So these are breathing frequencies that are typical of adult humans. This is a typical concentration of carbon dioxide in the air that we exhale. And then there are unit conversions that are applied. And basically what this tells us is that there's about a 14-fold difference in the mass rate at which carbon dioxide is emitted by an adult swimmer as compared to that same adult on a pool deck. So that has implications in terms of carbon dioxide that's present in the air. And we can account for that in the model. So again, I'm going to zip through this without boring you with the details of the 2-film model. But again, I'm happy to come back to it. So here are the final models that were developed for trichloramine and carbon dioxide. Let me just walk you through these terms. This term V sub G is the volume of air in the pool area. So imagine you have a swimming pool inside an auditorium. This represents the amount of air that's present, that volume of air above the pool. The next term describes how the concentration of either trichloramine or carbon dioxide changes as a function of time. Next, we have to account for outside air that's being brought into the facility. We need to account for the concentration of the volatile chemical that's present in the liquid phase. We need to account for the number of swimmers. And in the case of CO2, we need to account for the number of non-swimmers. So this term over here represents then the concentration of carbon dioxide in the ambient air that's relevant in the CO2 model. We need to develop a mass transfer coefficient for the baseline conditions. Those are the conditions when we don't have any swimmers in the pool, but we have water circulation just by moving the water through the treatment system. We need to account for mass transfer that's attributable to the swimmers. We do that with a mass transfer coefficient. And then we need to do measurements of the airflow rate through the HVAC system. So what we did was we worked with the people from the facility to conduct these measurements. And at this particular facility, what we did was we did measurements on all 5 of the air handling units that exist at this facility. So for each of these air handling units, which operate independently, we conduct measurements of airflow rate at each of the 3 damper settings. So these 3 damper settings that we applied were 30% open, 65% open, and 100% open. And the reason for doing that is that at this facility, they have real-time measurements of the damper opening as a function of time. So since we know the damper openings, what we can do is then use these measurements that we conducted to relate those damper openings to an airflow rate. So from this, now we have these relationships that allow us to estimate the volumetric flow rate of air into the system. And this is outside air that's being brought into the system. Okay, so on this slide, what I'm illustrating is how we fit the model to the data. So in this modeling exercise, we had measurements of all of the parameters in the model except the mass transfer coefficient. So what we did was we fit the model to the data using least squares analysis. So basically, we were looking for the value of the mass transfer coefficient, K prime, that minimized the error, or the, let's say, that optimized the fit of the model to the data. And we came up then with an estimate of the mass transfer coefficient by doing that. So this is an experiment where we did that with trichloramine. Here's a similar experiment where we did that with carbon dioxide. And again, what we're doing is looking for the value of the mass transfer coefficient that optimizes the fit of the model to the data. And if we separate out just the fit of the model to the data, what we observe is that the model does a pretty good job of replicating the actual dynamic behavior that was measured in this system. In addition, when we compare the mass transfer coefficient estimates that we came up with from these various experiments that are illustrated in the last two columns of this table, when we compare those with other mass transfer coefficients for similar systems that have been reported in the literature, what we find is that our estimates are consistent with those literature reports. So let me just walk you quickly through this table, and I'm not going to go through all of the data. All of the information presented above the red line corresponds to experiments where we focused on trichloramine. The information below the red line was in experiments where we focused on CO2. And I'm going to focus on the first two entries in this table. This value over here that I'm hovering over with the cursor, KLA, is this background mass transfer coefficient. So this describes the rate of transfer that's attributable to just circulation of water through the treatment system. This corresponding value of a mass transfer coefficient is the mass transfer coefficient that describes the transfer of a volatile chemical that's attributable to one swimmer. So what we learn from this and from the other experiments is that it doesn't take very many swimmers to overwhelm the mass transfer that would be observed just in the baseline case. And if you put several hundred swimmers in a pool, then the background or ambient mass transfer that takes place just by circulating water through the treatment system becomes negligible. So the swimmers play a dominant role in moving compounds from the liquid phase to the gas phase. So next, what we wanted to do was to apply this model to simulate indoor air quality under various conditions. So the model itself provides these quantitative links between the number of swimmers, the type of swimmer, and the indoor air quality, and it allows us to anticipate problems before they occur. So this would be a problem. We would expect bad air quality in a facility that looked like this for obvious reasons. So the idea here is that this model allows us then to simulate the implications of this condition or some other hypothetical condition on indoor air quality, and also allows us to evaluate the effects of various engineering interventions that we might use to improve air quality, like increasing the ventilation rate through the HVAC system, or incorporating an air stripping device like this one to improve air and water quality. So let me just walk you through quickly a couple simulations. So this is a hypothetical simulation, and let me just sort of illustrate what's going on. The bars represent what we anticipate as the bather load in this facility. So if you've been operating a facility for a period of time, you probably have a reasonable idea of how many swimmers to expect. And so we've done that for this particular facility. The red line represents how the HVAC system is being operated. This one being operated at about 1 air change per hour. And then the blue line is an assumed value of the liquid phase trichloramine concentration. For this particular simulation, we assumed 1/10 of a milligram per liter of trichloramine, which is typical for what we observe in these facilities. And then the black line represents what the model predicts in terms of the gas phase trichloramine concentration. Now, we're interested in maintaining that gas phase trichloramine concentration below some sort of guideline value. This is one of the guidelines that's out there in the literature. It was developed by the World Health Organization. They suggested 0.5 milligrams per cubic meter as a guideline. In other words, we want to stay below that. And what this simulation suggests is that there's going to be a period of a few hours in the late afternoon on this day where we're going to see trichloramine concentration considerably in excess of this World Health Organization guideline. And the model suggests not only how high it gets, but also how long it's going to persist. So what we observe from this simulation, again, is that these increases in trichloramine concentration are going to be strongly related to swimmer number. But now the facility management has a tool that they can use to predict this. And that tool can also inform control measures that we might use. So let's say that we have that same circumstance that I just described, where we know we're going to have a large population of swimmers over here. And one control measure that we might apply would be to just increase the number of air changes per hour for that period of time where the large number of swimmers are present. By doing that, what we observe is that we're able to keep the concentration of trichloramine below that guideline value. And we do it for this short period of time, recognizing that there is an expense associated with operating an HVAC system at 3 air changes per hour as opposed to 1. So the idea here is that this allows us to optimize the performance and let's say the management of that HVAC system. Alternatively, we could introduce an air stripping system. So in an air stripping system, what we're going to do is remove volatile chemicals from the liquid phase. Here, what I've assumed is that for a period of time, we're going to operate this air stripper for maybe 3 hours, and we're going to do that in anticipation of this large number of swimmers who are present in the pool. We'll assume for purposes of this simulation that that air stripper will reduce the concentration of trichloramine by half in the pool. That's just an assumption for this model. And the result of that is, again, we're able to stay below that World Health Organization guideline. So the idea is that this tool allows us to evaluate various interventions that we might use and also tells us when we should use them and how aggressively we should use them. All right, lastly, I just want to point out that this is a model. All models are simplifications of reality, and the user has the responsibility of recognizing the limitations and sources of error that are inherent in a model. So we've identified a partial list of known sources of error that are presented here, and there are clearly other things that are going to be sources of error. So as I mentioned, this is a 3-phase project. For phase 3, again, what we plan to do are conduct measurements and numerical simulations at other facilities. These would be in other geographic locations and would involve other facility types, including indoor splash parks and therapy pools. For each of these facilities, we have a list of requirements. These are basically things that we need to be able to apply the measurements and the model to these facilities. So we need to be able to quantify how much outdoor air is being brought in. We need to be able to measure how the gas phase concentrations of the volatile compounds of interest change as a function of time. And we also need to be able to conduct analogous measurements of those dynamics in the liquid phase. We need to quantify how many swimmers and non-swimmers are present in the pool. And we also need to be able to quantify operational characteristics of the water features, let's say in a splash park. So again, what we want to do is to extend this to other facility types, and that would involve developing estimates of mass transfer coefficient for other swimmer types and other, let's say, water features at splash parks, develop a user interface, and then allow for application of this model for real-time control of indoor air quality. To do this, again, what we want to do is to extend this to these other facility types and other locations. And one of the things that we want to do is to take advantage of an analytical method that we've recently developed and patented that allows us to measure aqueous or liquid phase trichloramine, and it gets rid of the MIMS instrument. So it doesn't require us to haul this MIMS instrument around to various pools. The basic idea is that we're going to take a sample, let's say in a flask over here. We're going to connect that sample to a peristaltic pump. We're going to pump that sample into this little vial. This vial will contain a membrane that will allow transfer of that compound into the liquid that exists in that vial. And we're going to put in that vial a liquid that contains indicators that will allow for measurement of trichloramine. So this colorimetric signal that we developed, that's probably very familiar to many of you, allows us to identify and quantify liquid phase trichloramine, and it's a really simple method to use. So I'll conclude by acknowledging the Council for the Model Aquatic Health Code and my collaborators, as well as the staffs of the participating pools. I want to point out also that there's a series of publications that are available now that describe this work in much more detail. The first was published in this journal called Water, which is an open access journal. Anybody can get this. The second was published in Environmental Science and Technology in 2021. And then just within the last couple of weeks, we had another paper accepted for publication in Building and Environment. And that paper is illustrated here. With that, I'll conclude, and I'll do my best to try to answer any questions that you might have. Thank you.
Well, first of all, thank you so much, Dr. Bletchley, for sharing all this with us. And thank you and your team for this incredible research that you have done. We're going to start here with some questions that we received in our Q&A box. I want to encourage everyone to continue submitting your questions. We might not get to it in the next 4 to 5 minutes, but we will share this with Dr. Blatchley after the webinar. And then if that's okay, we can put together a Q&A document to be shared with everyone. And as a reminder, you're going to receive a copy of the slides, the recording, and the Q&A questions and answers after this via email. So the very first question to get us started here, sometimes during some competitions, the pool circulation system is shut off. Was this taken into consideration? And if testing occur at times when sister circulation and when not circulating, was there any observation noted in regards to the circulation systems versus not?
Yeah, so the air circulation system in the facility that I focused on in this presentation, we were able to monitor its performance in real time. And it was never turned off in this particular facility, and I would never recommend that. I can't imagine a circumstance where you would want to do that. In fact, especially during periods where the pool is heavily occupied, that's when you're going to want to increase flow rate through the HVAC system. So I don't think it would be a good idea to turn this off during competitions. And if that's something that's actually being done, I would strongly suggest that we encourage people to change that practice, because that's potentially dangerous in terms of air quality.
Yeah, I would agree. And it says pool circulation system, so that would go for the water circulation within the pool itself as well.
Yeah, I'm not. I suspect that might be something where you know, the difference between first and second place in especially elite level competitions is often a pretty small distance or time. And I suspect that swimmers might get a little picky about air or water circulation in the pool and how that might influence their time. But even there, I think, I don't think there's huge benefit that really would be associated with that. And I wouldn't recommend that sort of practice.
All right, so our next question here is, are natatorium turnover rates available at these locations?
I assume that's referring to water turnover rate. And so, yes, they are available, and they typically operated as sort of in the standard range of 4 to 6 turnovers per day. I don't have the exact numbers with me, but yeah, I think they are available.
Thank you. Our next question asks, is it possible to calculate or estimate the atmospheric concentration of trichloramine if we measure the concentration of CO2?
Great question. So I think the whole idea, I'm going to bring up a slide and share that. So the idea of this slide is that there is a very strong correlation between the gas phase CO2 concentration and the gas phase trichloramine concentration. So that's evident in this slide. I think we could go further and say that it may be possible to estimate gas phase trichloramine and or control gas phase trichloramine by measurements of relative humidity. So these are complicated questions and they're not straightforward. So we don't have equivalent correlations for other facilities. We only have this sort of correlation for one facility. But the fact that these correlations are so strong, first of all, scientifically is not really that surprising. But I would feel a lot more comfortable about providing that as guidance if we had similar correlations at many other facilities, because there are other factors that we need to consider. Hopefully that answers your question.
Yes, it does. Dr. Blatchley, it's 3 p.m. here on my clock in the East Coast. So unfortunately, and to be respectful of your time and everyone's time, we'll have to stop here with the Q&A. But I want to say that we did take notes of all the questions that were submitted. We'll share with you via email, if that's okay. And then we'll put together a Q&A document that will be shared with the materials following this webinar. I do want to thank everyone for joining the webinar. As I mentioned, this was recorded. We will share the recording with a copy of the slides and the Q&A document afterwards. And we look forward to seeing you here in our next webinar. And I just want to say thank you again, Dr. Blatchley, for you and your team for this incredible research and for being here today. Have a great afternoon, everyone. Thank you.
You too.